Hello everybody. The title of my talk is Testing of Plant-Based COVID-19 Vaccine in Non-Human Primates in Thailand. I think as of today, everybody knows very well about COVID-19 vaccine. However, I will give you some more information. The COVID-19 was first reported in Wuhan, China in December 2019, and Thailand is the first country that made confirmation of the case outside China on January 13, 2020. After that, it has become a pandemic disease and quickly spread. As of June 2nd, about 170 million cases with 3.5 million deaths were confirmed. In total, about 1,200 million doses of vaccines have been administered, or if we counted that, one person should be fully vaccinated for 2 doses. It means that only 600 million people are fully vaccinated. And if we calculated from the world population of 7,900 million people, it means that only 13% of the world populations are fully vaccinated. If we take a look at Thailand, as of June 2nd, poorly, we have 165,462 cases and 1,107 deaths, but only 1.6% of Thai population are fully vaccinated with either AstraZeneca or Sinovac vaccine. This picture is to show you the process of vaccine development. Generally, to produce one vaccine, if we start from the R&D discovery, it will take about 10 to 15 years. But for COVID-19 vaccine, it can be amazingly happen within 12 to 18 months. If we look through the whole process of vaccine development, one of the important processes and very challenging for researchers is the step of preclinical trials in cells or laboratory animals. And for COVID-19 vaccine, we need mice or rats and monkeys for the test. Within Asia, there are very few primate centers, and luckily, we have one in Thailand, the National Primate Research Center of Thailand, or NPRCT. In the past year, the NPRCT has been involved in COVID-19 vaccine testing, and we are appear in many media in and out of Thailand. Allow me to give you the information of the NPRCT. Our center is accredited by AAA LAC International on February 26, 2020, and certified by OECD GLP on May 11, 2021. At present, we have 6 buildings, including 1 administrative building or A1, 4 breeding buildings or B1, B2, B3, and B4, 1 research building at biosafety level 1 and 2 or R1 building, and we are installing the mobile laboratory at biosafety level 3 or ABSL3 module. We now have 571 individuals of Pseudomonas macaques in our center. Since last year, we have tested the safety and immunogenicity of 3 types of COVID-19 vaccine in Pseudomonas monkeys, that is, mRNA, DNA, and plant-based protein subunit vaccine. For today's talk, I will focus on plant-based protein subunit. This slide shows the structure of SARS-CoV-2 virus and how it invades into the human lung and digestive cells. The virus uses spike protein or S protein, especially the receptor binding domain or RBD, to bind with ACE2 receptors of human cells. Then, for the plant-produced protein vaccine, it has been produced in the form of RBD conjugated with Fc region of an antibody and mixed with alum. Besides, we also mix two excipients with this vaccine, aiming to help stabilizing the vaccine at the storage temperature of 2 to 8 degrees Celsius. Because the cold chain transport is a big problem for distribution of vaccine to the rural areas. There are two excipients, sucrose and glycine. Because glycine, which is listed as food additives by the FDA, but no longer regards it as GLAS or generally recognized as safe, and the toxicity data are required to show that any proposed level of glycine use will be safe. So, for this experiment for today's talk, we will conduct two groups of treatment, vaccine group and excipient groups. This slide shows the vaccine production. The vaccine is provided and produced by the Bayer Phytopharm, which is a startup company under Chulalongkorn University. The production starts from inserting RBD-Fc genes into a grow bacterium, which will be infiltrated into Nicotiana bentamiana plant. Nicotiana bentamiana is a close relative to tobacco, which we say in Thai as Bayer. That is how the name of this company, Bayer Phytopharm, comes from. After infiltration, the plant will be kept for 4 days before harvesting. After the plant harvesting, it will be extracted for protein, purifying and analyzing, which you will get the vaccine, and here we name it as BY301 vaccine. For COVID-19 vaccine testing, theoretically, the COVID-19 vaccine testing for safety and immunogenicity test in monkeys will be done after the toxicity and immunogenicity test in rodents such as mice and rats show the positive results. After the safety test in monkeys, the vaccine will be entering into the clinical trials in humans for safety and immunogenicity test. And at the same time, the efficacy test in monkey can be done in parallel. Why? Monkeys are important animal models for COVID-19 vaccine efficacy test. On this slide, on your right-hand side, you will see the structure of SARS-CoV-2 in pink color and the ACE2 receptors in yellow color. SARS-CoV-2 virus binds with ACE2 receptor by using a spike protein. At the binding between SARS-CoV-2 spike protein and the ACE2 receptor at the human lung. If we zoom in at the binding regions, 5 amino acids at ACE2 receptor play a major role on binding affinity. From the publication of Luan et al. 2020, They researched in 42 mammal species for these 5 amino acids and binding affinity, and found that ACE2 of mice and rats cannot bind with the spike protein of SARS-CoV-2, but the monkeys can. This explains why monkeys are needed for efficacy tests of COVID-19 vaccine. Again, by the study of Damas et al. On July 31, 2020, they looked at 25 ACE2 amino acid residues that bind with spike protein. In total, 410 species of vertebrates, from non-human primates to amphibians. They found that 18 species of oval monkeys and great apes have all 25 ACE2 amino acid residues identical to that of humans. Non-human primates are identified as very high risk for SARS-CoV-2 infection and become the most suitable animal models for COVID vaccine research on efficacy tests. This slide shows the experimental protocol for this study. We used juvenile female xenomorphous monkeys, 5 monkeys in each group, for 2 groups of excipient and epv2021 vaccine at a dose of 10 micrograms. The excipient and vaccine are intramuscularly injected at the quadriceps femoris muscle of the monkey's leg on day 0 and day 21, or 3 weeks apart. Blood collection and safety tests are conducted on day 0, day 14, and day 35, or 2 weeks after vaccine injection. In our primate center, we conducted the safety test in compliance with the OECD GLP principle, following the guideline of ICH S7A, ICH M3R2, and WHO Technical Series number 927. We observed 3 vital organ systems for safety pharmacology, that is, cardiovascular system, central nervous system, and respiratory system. This slide shows the safety pharmacology test for blood biochemistry on day 0, day 14, and day 35. On the slide, green color is for excipient group and orange color is for vaccine group. So you can see that no significant differences are detected between day 0 and day 14, and between day 0 and day 35 in both excipient and epv2021 vaccine group, except for blood urine nitrogen, or BUN, of epv2021 group, that day 14 is slightly lower than day 0. This slide shows the safety pharmacology test for hematology. So you can see that no significant differences are detected between day 0 and day 14, and between day 0 and day 35 in both excipient and epv2021 vaccine group, except for number of eosinophil, or EO, of excipient group, that day 14 is slightly higher than day 0, that you can see the light blue asterisk indicated. This slide shows the safety pharmacology test for physical condition, central nervous system, and rectal temperature on day 0, day 14, and day 35. For physical condition, we observe the appetite of monkeys, or food and water consumption, and fecal condition. For central nervous system, we observe motor activity, behavioral changes, coordination and sensory motor reflex responses. All parameters are normal. And monkeys also show normal rectal temperature. So you could see that the normal range of rectal temperature in monkeys is a bit higher than human. That is 36 to 39 degrees Celsius. This slide shows the safety pharmacology test for cardiovascular system and respiratory system. We determine blood pressure, heart rate, and electrocardiogram for cardiovascular system, and respiratory rate, hemoglobin oxygen saturation, and lung sound for respiratory system. So you can see from the graph, all parameters are normal. Besides, no adverse reaction at the injection site, including edema and redness, was observed. Overall, you can see that 2 times of intramuscular administration of the excipient and BY301 vaccine are unlikely to cause adverse effects to the physiological function of 3 vital organ systems, including central nervous system, cardiovascular system, and respiratory system, which reflect with relevance to safety in healthy pubertal humans, consistent with the scope of the ICH S7A in safety pharmacology studies for human pharmaceuticals. This slide shows the immunogenicity study, a 50% neutralization titer, or MN50. On the left-hand side, it is a graph for excipient group. You can see that the MN50 titers in all 3 days, day 0, day 14, and day 35, are lower than the limit of detection, or LOD. On the right-hand side, it is a graph of the BY301 vaccine. You can see that the MN50 titer in day 35, or 14 days after the second immunization, is significantly higher than the positive control serum. This indicates that the BY301 vaccine is promising for stimulating the immune response and ready for clinical trial phase 1 in human and for efficacy test in monkeys. Then, the BY301 vaccine from Bayer Phytopharm Company will move to the efficacy non-clinical test in monkeys and clinical trial phase 1 in human in the middle of this year. Since our primate center experienced testing of various kinds of COVID-19 vaccine, we will hold the one-day hybrid symposium at the theme of COVID-19 vaccine in Thailand and the role of laboratory animals, learns from experiences, Please access into worldwideweb.nprct.org for more information. Thank you.